Okay, good afternoon, good morning, good evening from wherever you're joining us. I would like to welcome you to the education workshop, Lessons Learned in Implementing Inclusive Online Labs. My name is Anna Allen, and I am one of the chairs of this workshop, and I'm from Howard University in Washington, D.C. And my co-chair is Edward Freeman from St. John Fisher College. In this session, what we're going to do is we're going to have a keynote talk by Dr. Alana O'Reilly. It'll be about 20 minutes long. And then we're going to have 3 selected talks that have been selected from abstracts submitted, which will each be 7 minutes long. We're going to ask and take all questions at the end, but we ask that during the session that you can enter your questions in the Zoom Q&A box. And then some of the speakers might choose to start to answer some of those directly through the Q&A feature. But then we should have about a half an hour at the end for hopefully what will be a lively discussion. So with that, I'd like to introduce our first speaker, the keynote talk being given by Dr. Alana O'Reilly. She is from Fox Chase Cancer Center and founder of the eClose Institute. And so Alana, it's all yours.
Hey, thanks everybody and welcome. I love SDB and always have. So my talk is about power to the participants. And so what I'm going to try to do today is to have everybody engage in your own personal power and how that contributes to science. So what I want to focus on mostly is the why. Why do we do science? Many of us have our sort of canned expected responses from the scientific community. I've loved science since childhood. I'm curious. I love to solve puzzles. I want to help people, or my parents think I'll be a really good scientist. And why cures? Why do we care about giving undergraduates, or even in our case, younger students, in immersive research experiences? And that's because unambiguous evidence has shown that research experiences connect pieces of puzzles that create a rising vortex for student success in STEM. Why inclusion? People say now, especially in the past year, like the diversity of thought is what drives innovation and integration of science into life. So what I want to think about today is these are the sort of answers we're expected to give. It's like the code of science. We see this in our applications for our programs all the time. I've been curious about science since I was an infant. And that's true of all humans. So why are you doing science now? Science is so deeply personal. I want everybody to really take a hard look in this mirror and say, really deep down, why am I doing this? Discover your vulnerability, which is a major advantage of doing online learning, because you can be anonymously vulnerable, right? Really dig deep into the vulnerabilities that may be able to enhance not only your science, but that of your community. So I'm hoping is to be done early. And so if you have a second device, you can go to joinpd.com and type in this code, and we'll have a little activity at the end if there's enough time. Oh, I forgot to say, I put this like on every slide just in case you missed it. It's here in the corner. So what we try to do in our programs is first to identify the purpose of our participants and then honor that. And so this is a video from an undergraduate who participated in our program last year. Depending on which Zoom you use, it switches over to YouTube, but anyway.
So coming into this project, I was most interested in studying squamous cell carcinoma, which is a type of skin cancer that affects over 1 million people in the US every year. One of those people was my grandmother, who I lost to the disease in 2015. You might notice a small blue butterfly floating around for my presentation. That's my Zammy. She'll be joining us today.
So one of our most important drivers is to create a program where students come in with their Zammy or whatever it is that matters most to them and their reasons for doing science, and allow them to celebrate their purpose. So why is this important? It's important for the individual unambiguously, but it's important for everyone. So what we have found is that retention of diverse people in science may really depend on the why. And the why is something that we have to start to investigate. So why are we losing so many underrepresented minorities who earn their Bachelor of Science degree? And it's not even really that many. I have to take that back. It's only 16%. But by the time we get to full professor, we've made exactly 0 progress in increasing full professor appointments for underrepresented minorities. in over a decade. So what are we going to do about this? Are we basically causing people to code into our prevailing scientific viewpoints, or are we allowing them to celebrate their why? So this is also a primary driver of health disparities. This is an example of the impacts of deaths for COVID-19. And you can see that underrepresented minorities have an egregiously higher incidence of death. This is also mirrored in all of these other diseases: cardiovascular disease, hypertension, diabetes, and cancer, and has been directly linked to the dearth of underrepresented scientists in research. So how do we take our why, the scientist why, and connect it to the community why or the students why in order to create projects that actually make impact? And so this is basically my why. I'm really interested in signal transduction. We found out several years ago that the single nutrient cholesterol triggers off hedgehog signaling to control a single cell in the fly's ovary called follicle stem cell to affect its function. And so this observation led to a hypothesis that every component of a diet impacts one of these little proteins in this massive Drosophila signaling integrative diagram to affect a cellular response, and that the collective response when somebody eats leads to the organ appropriate organ function. And so this is an enormous problem if we want to understand how each and every nutrient affects each and every protein. So we need a lot of help. So why would this be interesting to young people? They're athletes, they're interested in diet. Many of their friends have body image or eating disorders. They're interested in preventing diabetes, and they've observed adults in their life who maybe have cancer. So this is a topic that's extremely engaging to pretty much everyone with the overall question of what should I eat? So what we try to do is make it real in that sense, and also make it mine. How do we allow a student to do what belongs to them in the context of actually pushing science forward in a rigorous way? And so what we try to do is increase research literacy through student-centered active learning in a transdisciplinary way to increase their self-efficacy and instructor and student agency. So what we developed is a program that was originally called Kids Conquering Cancer, because I work at Fox Chase Cancer Center. Now it's Kids Conquering Cancer, et cetera, because the students come in with whatever primary health concern is major in their community. And so we've created a series of iterative level programs. All of these levels occur prior to high school graduation. So what we're going to talk about most today is the undergraduate STEM. And so students are investigating how the 1 million different nutrients impact 300 cancer genes or disease genes from other diseases. And even just that is 300 million single combinations. So it's something that's essentially an infinite problem for contribution to be made. So how do we do this? We use Drosophila as a model system where we very rigorously know the full life cycle, full development cycle. It's inexpensive. It's safe to use at home, and we use flies that are either wild type or bear mutations in oncogene, tumor suppressor genes, or other disease-causing mutations. Mostly we screen with dietary supplements, again, because they're inexpensive and safe at home, but students can also bring in compounds from their cultural backgrounds or their life experience to test to try to see what's going on. And so the way it works in our undergraduate bridge program is that a series of students will screen mutants in sequential mutants in a pathway in order to identify where in the pathway a particular compound works. So our original one we identified was selenomethionine, which affects a protein called corkscrew that's in the RAS pathway. And so Sammy and Gabby discovered that grapeseed extract inhibits RAF. And so the other students in the program have found specific nutrient compounds that inhibit AKT or again, have an opposite effect on corkscrew compared to selenomethionine. And these are the young ladies that have been the primary drivers, but we've actually had almost 1,800 students contribute to this EGF receptor screening project. So after this, right, that was sort of my thing, like, okay, we have the signal transduction pathway that we're interested in. What do the students want to do? Last summer, we switched the whole program to virtual. So all of the students received a research station at home, which I'll show you in a second. Claudia from California was interested in COVID-19, having a family who had been dramatically impacted by that. And she was interested in oxygen availability between wild type and AKT mutants, which are linked to poor outcomes in COVID-19. Adriana was interested in innate immunity, having family members with autoimmune disease. Danish has a dad who has type 2 diabetes, and he wanted to understand if he could find nutrients that might be helpful to his dad. Gilharia is interested in pain sensitivity, particularly in patients with cervical cancer, having had relatives, again, who experienced extreme pain. And Kiki is interested in breast cancer metastasis using the fly ovary border cell migration assay to investigate how nutrients intersected with CDK4 signaling pathways to affect metastatic processes. And most of our students from Philadelphia actually focus on breast cancer because the incidence for young women with breast cancer is extremely high. Oops. So at the end of the day, we really trust our students to come up with important, impactful projects. And so what they tell us is that this is all about trust. You're the first people who believed in me. And so this is Nicole Harrington, one of our, I mean, out of the park, write her name down. She is an out of the park scientist, and she has done molecular analyses. And then last summer she came and said she wanted to investigate how trust was impacting vaccine uptake in racial and ethnic minorities. She managed to publish a paper in less than 3 months and has now inspired another generation of our students to take a social behavioral approach to their research questions and really investigate those vulnerabilities that they've experienced in their own lives. So does it work? Our virtual hybrid undergraduate program from last year, 42% of the students said they definitely want to be a scientist, 43 said maybe, but every single student wants to do more research. And so this is something maybe we can talk about in the workshop is how do students maybe want to be a scientist or not want to be a scientist, but all of them want to do research. So somehow our definition of what being a scientist means is disconnected from doing research, which they truly love. Other metrics that we have in Philadelphia, 77% of college students switch out of STEM majors, but 72% of our students continue getting paid research jobs in college. 6% of inner city students in Philadelphia complete STEM degrees, and 100% of our students completed their STEM degrees on time. So this is a really impactful approach. And so how did the virtual part impact us? First, it induced panic. And then we said, all right, we just have to figure out how to recreate a research lab in kids' kitchens, bedrooms, coffee tables. And so this is the setup that we send them at home. And you can see there's pretty much a vortex, balance, little stir plate, everything that you would see in your professional research lab, but it's in their home. And this is John, one of our undergraduates from last year, modeling his setup here. So another way to unlimit access is that using Zoom, you can do real-time assessment and perhaps more importantly, assurance. So the students can hold up their vials, dip their camera down so we can see you have the right thing, you're doing it the right way, and tell them, you're doing great, like you got this, okay? You're making huge progress. We also have been able to unlimit access through Zoom by becoming geographically unlimited. And so we've been able to have students from many states across the country who are now all collaborating together in a political environment that is so charged, but now all of a sudden you have all these students collaborating to solve problems that matter to themselves and their families. So this was from this morning. We have an ongoing program with 81 students with the American Cancer Society. And this was, you get these really big rewards from students. I'm so happy I joined the program. It taught me a lot and it's fun, right? Research is fun. And that's, I think, one of the reasons that most of us use as our why, like we love this, right? And so now we're trying to instill this love in younger students. And then just as an ending point, the impact of the program is sort of like we were putting this slide together. And I mean, these are publications from students that we've had in our programs since 2013. And every single one of these publications occurred prior to college graduation. And so early research is experiences that give the students ownership and integrate with their family needs really have a tremendous impact on their ability to conduct professional science. Oops. So these have been the people who funded us. And I really want to point out SDB, who was the first organization that funded any aspect of our program. And without SDB, there would be no program. So we are eternally grateful. This is the team. This is Darawice Whalen. She's a genius in terms of curriculum development for research programs, particularly virtually. These are our teachers and our team, most of whom are students who participated in our prior programs who we hire to help us going forward. So now I wanted to do that activity if we have some time. So I think we have plenty of time. So if everybody can go to joinpd.com and type in this code, we're going to have a little exercise to see what ideas you have about connecting your why, or even just, taking advantage of that anonymous vulnerability moment to share your why, and then connect that to the needs of your community or a community that your university institution is interested in supporting in STEM. And let's see if we can come up with some projects that resonate with those. So I'll just wait a second before I switch over. Does anyone want to pop on and tell me, I got it, I'm in?
I got it, I'm in, Alana.
Okay, thanks. Okay, so now I have to figure out how to switch to that. Okay, all right, starting class. Okay, so now you should be able to just type in your ideas. I have to show responses. Okay, here we go. Yay, motivated to get students excited about science and everyone to feel like they have a place. Okay, and this is exactly what we all want, I think. So how do we do it, right? So how do you take that why and make it practical enough to engage your...
Alana, I think we're still seeing your PowerPoint presentation.
Okay, all right, wait, so, okay. So I'm gonna stop sharing and then reshare, okay? When I did this on my own thing, it worked. Okay, wait, how do I get rid of this? Sorry, everybody. Okay, resharing. Where is it here? Okay, because I can see it. It looks awesome. Okay.
Now you're good. Now we can.
All right, here we are. Okay, so we want everyone to have a place. Enjoy doing science with a community of people. Good. Excited about discovery and training the next generation. Just like to play in the lab. Flies are awesome models. Okay. Yeah, so yeah, I think it's really interesting. Like the students that we get, for example, we had one student who came because her mother had breast cancer, and she wanted to find a solution to keep her mom alive. And so, yeah, so I think it's really interesting how young people, particularly from inner city communities, have sort of a different why, right? They have a driven why, purposeful why that's very, very personal. So yeah, so does anybody have like what their science is right now and how they think they could connect to creating a project with the community? So how could you take your love of flies and engage the community around your institution? Oh, surprises, yeah, I love those too. Okay, yeah, motivate students to think critically. And that, right, this is a key thing. Like, how do we integrate sort of the biology that we're mandated to teach by our university curriculum, or in our case, high school and middle school curricula? How do we use that to help students understand how to make these decisions in their daily life? Like, are we using language that they can use to talk to their doctor? Are we using language that they can use to explain to their parents how COVID vaccines work? Because a lot of parents are reluctant to have their children vaccinated. So how do we as scientists really connect to the real world? Yeah, this kind of idea, right? Help students sort out facts-based news versus fake news. That's really, really important. And it's so prevalent right now that people are more using things like Facebook to understand how to make themselves healthy, right? Rather than us, who are the scientists. So much is unknown. Yeah, that's true. Yeah, and share with the community. So if you put this, how do you share with the community? Like, I think that's something that we're here for, right? For the workshop, like how do you like to do that? Yeah, young people see that we don't know. They're always surprised about that in our program too. they say, well, what's the answer? We're like, you got to tell us. We just don't know. Yep, science is power. Recruit and retain, yes. And that's exactly what we're trying to do. And I'd like to know also what you think about our hypothesis, like retaining the why. Spread love of curiosity for sure. Spark. Dev bio is definitely the prettiest by far. When you do a lot of talks like this, it's you know, you get people from a lot of different things, but it's so true that development is amazing. Okay, yeah, so congenital defects, and we've actually had a number of students who've had spina bifida, or one student had a brain anomaly that had to have surgery early on, and those students, you know, they are basically taking the power into their own hands of trying to figure out how to, you know, how to solve their own health problems.
1 minute left, Alana.
Yep, yeah, we can stop whenever you're ready. You know, this is where, yeah. Yep, kids can do science, that's for sure. And then somehow school teaches them they can't, right? And then we have to kind of like recruit them back. So how can we make this a trickle down solution so that young kids don't ever leave the science, that they love it all along? Yes, embryos developing. When I was a graduate student, the first time I saw a Xenopus embryo divide, I almost passed out with joy. Science art. Okay, yeah, that's an interesting one too. That's has become more popular, and yeah, so I'd love to hear more about how that can be used to really celebrate the why. Schools and teachers, yep, trusting.
Okay. So I think with that, we're going to move on to our next session, but I want to thank you so much, Alana, for getting our conversation started and everything. And I'm sure I can see questions starting to pop up in the Q&A. So that's my reminder. If you have a question for Alana, please put it in the Q&A, and some of them we will be answering live and having the discussion, and others we will be answering via the Q&A feature. So with that, I'm going to turn it over to my co-chair, Ed Freeman, who's going to handle the rest of the instructions.
Thanks, Anna, and thank you, Alana. It was really wonderful to see and hear your enthusiasm for teaching a younger generation that they can in fact do science. That was great. The next three speakers are ones that the Professional Development and Education Committee chose from actually a large number of submissions. So we were really pleased with the overall number of submissions, and we had some really stiff competition. But what we wanted to do was try to hear from people doing some different things. So we're going to start out by hearing from Isha Verma, and she's coming to us, if I'm not mistaken, from the University of Michigan. Is that right, Isha?
Yes.
Okay. So you'll have 7 minutes, and at the 1 minute mark, I will give you a 1 minute warning. Thank you.
Can you see my screen?
Yes.
Okay, thank you. So good afternoon, everyone. I want to thank the organizer for giving Developing Future Biologists an opportunity to present our education and outreach work here at the STB annual meeting. In my talk, I'm going to focus on how we converted our hands-on laboratory short course into a wet lab at home experience. Just to give you a brief introduction, Developing Future Biologists, or DFB in short, is an educational outreach organization at the University of Michigan, Ann Arbor. Our mission is to ensure that the next generation of biologists, regardless of race, gender, or socioeconomic structure, socioeconomic status, can learn the core concept of developmental biology and are made aware of the different career opportunities in the area of science. DFA organizes a week-long summer short course aimed at teaching the undergraduate student the fundamental concept in developmental biology. DFB members include faculty, postdocs, and graduate students. DFB is mainly led by postdocs and graduate students who design and teach the lab and coordinate with the faculty members. The teaching assistant for the course are selected from the DFB student alumni. This unique trainee-led structure provides additional opportunities for peer mentoring, teaching experience, and science education. The inaugural course of DFB was held in 2015. From 2015 to 2019, we conducted this incredibly engaging in-person wet lab experience for our students. However, when the pandemic hit, we had to transition our course to a virtual format for 2020 and 2021. We tried to provide this engaging hands-on experience for our students despite not being able to teach them in person. So for this, we designed a virtual 1-week-long course that included multiple hands-on labs that focused on various model organisms, including planarians, zebrafish, mouse embryos, and Drosophila. During the course, we also engaged the students in bioinformatics, PyMol, and ImageJ labs. Our faculty member provided lectures on fundamental developmental biology concepts. We also conducted multiple professional development workshops and panels during the course. While it was easy to conduct the professional development workshop and panel in the virtual format, it was a bit challenging to provide the hands-on lab experience in the virtual format. For this, we designed the lab activities that can be performed by the students at their home. Each student was supplied with a lab kit. Each kit included 2 microscopes, a USB-powered pluggable microscope for gross anatomy, and a paper fold scope for large magnification viewing. We provided students with living organisms, which include planarians and Drosophila. Planarians were used as a simple and effective means of observing regeneration. Drosophila was used as a part of lesson on disease and development. Students performed the dissection of ovaries and intestine on Drosophila and observed eye tumors. We also supplied mouse embryos and embryonic skeletal prep and zebrafish embryos for the embryology lab. To ensure that our students are successful in conducting the labs at their home, we provided the students with lab supplies a couple of weeks ahead of the course and also initiated our engagement with the student weeks before the course. We conducted pre-course information session where we demonstrated the students how to use the microscope and answered any questions that they have. We divided the students into small group of 3 to 4 and assigned 1 instructor per group so that the student can start to engage with the instructors and other group members before the course. These are some of the images that are collected during the experimentation by our students of Drosophila, planaria, blood smear, mouse skeletal prep, and zebrafish embryos. Most of our students were able to successfully complete the lab activities at their home. One of the main focuses of the DFB is to promote inclusivity and diversity in the field of developmental biology. For this, we carefully design all our lab activities to cultivate a sense of academic belonging in the students. We recruited our students from underserved and diverse backgrounds. We reached out to faculty at multiple schools, including tribal and community colleges throughout the US, and asked them to encourage their students to apply. During our course, we highlighted the diverse range of scientists working in the field of developmental biology to promote the student sense of connection to the field. We intentionally recruited diverse course instructor and the panelists for our course. We provided clear communication to our students through multiple channels. We provided a course handbook that contained all the course norms and expectations. The handbook also provided all the information that was required to conduct this for the student to conduct the experiments at their home. We used the Canvas module for other students to provide all the resources in one place. Additionally, we used discussion board and Slack for the student to promote their participation and for them to share their images, question, and provide feedback. We also tried to facilitate structured interaction between the students. As I mentioned earlier, we divided the student into a group of 3 and 4. We carefully designed each group to leverage diversity and avoid isolating the student with underrepresented identities. Students in small groups work together on brief, well-defined activities under the supervision of their group leader and discuss their result among their small group as well as with the students in other groups. We also incorporated accessible learning strategies. In order to overcome the financial barrier that is faced by many students, we provided all the course supply free of cost and also loaned laptop to students in need. We developed alternatives for living organism-based lab for the students who were not comfortable with working with living organisms. Additionally, we recorded all the course activities and incorporated multiple learning methods such as interactive lecture, group discussion, and method board. As many of our students had English as a second language, this really helped to alleviate some of the discomforts that can come from speaking a non-native language.
1 minute remaining, Isha.
Sure. We conducted pre and post-course exam and surveys and feedback form to modify our course to better meet the student need and to assess the effectiveness of our course. For most of our students, we observed an increase in the knowledge of developmental biology after ending the course. student also reported an increase in the interest in the developmental biology, more familiarity with the career in science, and the likelihood of applying to the graduate school. Our goal is to help these students to become familiar about the career in science and get them excited about pursuing a science-related career. In order to do this, we have now started long-term mentoring. For this, we have started new initiatives like year-round programming, DFB Research Fellowship, as well as Conference Award. Altogether, these initiatives will significantly strengthen our alumni network and provide further support and opportunities to our participants well after the course has ended. We are extremely grateful to our sponsor who make it possible for us to provide the course free of cost to all our students. These are our contact details. Please reach out to us if you want to know more about our organization or want to collaborate with us. Thank you.
Thank you so much. Our next speaker is coming to us from the LaGuardia Community College, Thomas Onorato.
Okay. I'd like to thank, good afternoon, and I'd like to thank the co-chairs for inviting me to give this talk today. Today, I'm going to talk to you about how I've tried to democratically sequence cells to shift the paradigm of community colleges from consumers to authentic producers of knowledge. So I want to tell you a story that starts with a long-term collaboration between the Wessel Lab at Brown University and me at LaGuardia and the group I developed, the URGE, and how we became Primo and the URGE. And what we like to do with the URGE, the undergraduate research group on echinoderms, and we're trying to take the apprenticeship model, but move it further into the classroom to impact more students. And I do this in my cell biology class at LaGuardia. And so we want to try to shift this paradigm. And by having research institutions closely interact with community college faculty doing research, this could then hopefully impact community college students. And so by exciting research institutions, they could excite the community college faculty, which will excite the students and help bring them to obtaining their associate's degree and advanced degrees in STEM. And so this, we did this in wet lab, and then we decided to bring in computational biology during the pandemic to enhance the online component of the labs. And so the class that I've incorporated in online, this work into is the cell biology capstone for the biology program at LaGuardia. The platform was Blackboard Collaborate with synchronous online sessions. The lab assignments that we adopted for online was a mock NSF research proposal that students write in groups. So in spring 2020, we went down the rabbit hole of COVID chaos. The proposal was basically based on literature that was relevant to the URGE research interest. But then we decided with the Wessel Lab and we decided to bring in computational biology. And so we entered the wonderland of bidirectional learning, since this was new for me as well as my students. And then in spring 2021, we've realized the Mad Hatter is not so mad after all, and we enhanced from what we learned in fall 2020. So some highlights of this collaboration. In fall 2020, Natalie and Onorato from the Wessel Lab guest lectured to explain the project. And then students basically interacted within their groups. But in spring 2020, we used the previously recorded materials to overview the project. Natalie came back 2 more times to explain our studio software that students could use for the single cell sequencing analysis. And then students in all sections presented results and received feedback from the Wessel Lab members, as well as they provided feedback on their draft research proposals. So the single cell RNA sequencing assignment. The Wessel Lab had previously isolated cells from the C-star ovary. They used 10x genomics platform to do this single cell RNA-seq. And then they had produced data of producing 12 clusters of cells, populations of cells expressing similar gene expression profiles. So that X cell that came from the Wessel Lab and Natalie, my students in cell bio, we then started to annotate and do the bioinformatics analysis on this. And so this is what the X cell looked like. You had the transcript numbers and all this jargon. And then the students ended up getting several transcripts in their groups that they would have to annotate and analyze. So they first searched the Equino base with the transcript number. Then they would take the common name, any synonyms, and they looked at the translated peptide sequence to do further analyses. And they put this all in an Excel. And then they did protein blast to determine any homology that's known, what to give them a hint of what this transcript, what gene it might be associated with. And then they did PFAM analysis to see if there are any other conserved domains and so on. They did ProSite analysis to look at any consensus sites, any other domains, and any possible functions, as well as search the literature for possible functions for these genes the transcript might be associated with. So the student work. So here's an example of the students' Excel that they produced from the fall 2020. So here we see the annotations, their PFAM results. So even though this had no annotation, they still, by doing further analysis, saw that it could be associated with this gene. Then in spring 2021, we advanced it to introduce RStudio software. So students made feature plots and violin plots showing the gene expression in cells of each cluster. They also compared violin plots to look at the different genes and so on in each cluster. And then they did additional analysis where they can compare two genes and see how they related an expression in the same cluster as well as different clusters. And so here's an example of what the students concluded in their after doing all of this. They saw that in cluster 10, there was a lot of histone genes that were associated with this cluster of cells. And they saw synthesis of RNA, DNA, sugar nucleotides. And then they concluded that this cluster may represent oocytes from this sequencing of the cells in the ovary. So democratization and inclusiveness, the student impact. So after the year was over, we conducted survey of student responses. And we can see here that a majority of the students, and had about an N of 37 responses to this survey, that 70% were female, and a good majority of the students were from underrepresented groups.
1 minute remaining, Thomas.
And so when the students were asked, did you previously conduct or concurrently conducting any research? Almost 87% said they have never done this. And this is the important student impact, to democratize research. We're expanding beyond the apprenticeship model that's limited in number, and we're giving research opportunities that are authentic to a larger population of students. And they said, if the course goes back in person, would you keep the lab? And 54% said they would keep it in person, but with the bioinformatic component, showing that this was well received by students and something to further expand on in my future courses. And I would like to thank you for listening, and I'll take any questions during the Q&A. Sorry for talking really fast. I'm a native New Yorker.
Thank you, Thomas. That was great.
And stop sharing.
Our third speaker this afternoon, Jen, I never confirmed how to say your last name, so I'm not going to try. Jen's coming to us from Bryn Mawr College. Thank you.
Hi, yes, thank you. Jen Skirkanich is my name. So I'm just going to go ahead and, all right. Can everyone see that okay? Okay. So thank you so much for having me come today. I'm going to tell you a little bit about one lab module that I did this past year, which involved measuring tree biomass and mapping that across the community. When I say community, I mean my classroom community, which included both in-person and remote students. And the reason I'm talking to you about this today is because we were able to do this both in the classroom and with students that were remote, but also because this is something where the technique itself, which is a forestry-based technique, is able to be connected to environmental justice issues in our neighboring Philadelphia. And so this was really impactful in ways that I hope that you'll see. And so I just want to establish right away that, in thinking about inclusive classroom spaces in my laboratory, this is not really something that we do like on a module by module basis. It's really part of not just the content, but also the affective experience of being in the classroom. And so that's always a good thing to remember. And I'm just going to share with you two simple interventions that actually work really, really well in a remote space. And these are values affirmation interventions and utility value information interventions. So both of these are designed to mitigate the effects of stereotype threat that are measurable in the literature, and you can see actually the effect of this on the performance gap that people talk about quite frequently. And so a values affirmation intervention is one where you ask the student to just talk about something that's important to them. So they're given a writing assignment that was just spend 10 minutes thinking and writing about something that is important to you personally, anything that they feel comfortable sharing with the teaching team. And the types of things that I got from students from this assignment were really incredible and allowed me to connect with the students in a way that I really couldn't have in the online setting, in the Zoom room setting. And it also allowed them to know that I genuinely care about like things that are important to them as well. So utility value interventions are a little bit different in that they ask students to align something that they personally value with the core content of the course and ask them to reflect on that and also possibly reflect on the future utility or value of that subject to something that's important to them in the future. And so this was just to ask them to talk about how their life journey has intersected with elements of science. And this is after having a classroom conversation about expectations of the course and what science is, and just spend 10 minutes writing about that. Both of these types of interventions are great for online or in-person types of assignments and can really help students feel more welcomed in the classroom environment, but also help you connect with them a little bit better as instructors. So a little bit about the context of the course. So this is an introductory biology laboratory at a small women's liberal arts college just outside of Philadelphia. I have 3 sections of in-person learning and 1 section that is remote due to COVID this past year. Normally they're all in person and there's about 30 people in the remote section. So for this particular module, students are learning about photosynthesis, about the carbon cycle, greenhouse effect, effects of carbon dioxide and global warming, and about carbon sequestration in trees through photosynthesis. And they also learn a forestry technique that's very important in forestry research called the DBH technique to measure tree biomass. And then we further go on to talk a little bit about the idea of why you would want to know tree biomass and map the locations of biomass and how that's connected to particular issues in the city of Philadelphia. And so this is a really simple technique, DBH or diameter at breast height in forestry. It's a technique using a measuring tape where you simply wrap it around a tree. This is a special measuring tape that when you wrap it around the circumference of the tree, it actually tells you the diameter of the tree. But you can actually use anything for this. I actually have students use phone chargers, all sorts of stuff like that when doing this technique. And I made some videos to demonstrate using anything you can find to measure the circumference of a tree. And so in the forestry industry, they've actually taken a lot of empirical data about the above ground biomass in relation to the diameter of trees of particular species. And what they found is that there's a relationship that you can predict between diameter and biomass. And this is really useful to find merchantable lumber in a given unit of land for the logging industry. And what's nice about this is it actually develops these really simple allometric equations that allow us to just plug in DBH and find out the mass of the tree. And so once we have biomass, we can calculate the amount of carbon sequestered in the tree based on the knowledge of the carbon content of typical wood. And then students would then go ahead and use this Google Maps document that we have to note their tree, to pin their tree, and then also maybe share the biomass and a photo. And so we actually got this really nice kind of like class photograph, if you will, of all of the trees of our class. A lot of people around the Bryn Mawr area, especially for the in-person students. And this isn't live, so I can't zoom in, but it's just a snapshot of the class photo we took of students from all across the country and their trees. So why would we do this? Why is it important to know the biomass and map biomass of trees? And so we talk a little bit about forest inventories, and I will tell you, I am a developmental biologist and not a forest ecologist, but I've worked with forest ecologists and I've got to learn a lot about this. And so there's a lot of reasons why you would want to do a forest industry. In cities, the urban forest is basically the collection of trees in the city, and this is Philadelphia, where I live. And so why is it important? So trees are important for air quality, mental health, temperature regulation, physical health, and stormwater runoff mitigation, in addition to other important things that are important for the health of people in the city and the health of the city. And what we know is that trees are inequitably distributed across Philadelphia. And several studies have shown actually that tree distribution is tightly correlated to generational wealth that has been impacted by the insidious effects of redlining and how that still has repercussions for today. So for example, in redlined districts where individuals were, BIPOC individuals were prevented from living historically, we have an overlay here of the red line districts that contain much fewer amounts of trees compared to other areas of the city. We also know that redlining has this impact via trees of heat sensitivity. So we've got about a 20-degree difference on some days between these hottest areas of the city and the other areas just adjacent to it. And this has really extreme health effects on individuals in these areas. So there are a lot of issues about trees and tree distribution that are directly related to health and inequitable distribution of resources.
Another 1 minute remaining, Jen.
Great, thanks. Another impact that this has directly in the city of Philadelphia is the fact that Philadelphia has a lot of impermeable surfaces. And we know that when there's trees and other permeable surfaces, we have a lot of storm water absorption into the ground. However, in the city of Philadelphia, we have a lot of runoff. And you can see this is actually my house on Monday. I live in a row house in Philadelphia, and this was what the roof looked like when we had a storm. Lots and lots of impermeable surface here and water running right off of it, right down the street, and water kind of in these rivers going into the sewer system, which, you know, you might think that's fine. But what you may not know about Philadelphia and similarly aged cities is that the sewer system cannot actually contain all of that water. We have more water in rain events, and we have more impermeable surfaces. And so what we actually end up having is something called a combined sewer overflow. And that's when raw sewage from sinks and toilets and houses combines with that water and overflows directly into the river system. And this has been a huge problem. About every 6 days, raw sewage is dumped into the sewer systems in the Philadelphia area. And this is sewer system. These are areas where kids really like to play and people go fishing all the time. And so this is something that really impacts the health of our city. And you could even see they have an app where you can actually look at each of those dots is one of those combined sewer overflows. This is right after I took that video. A few were overflowing. And you can see that they begin to turn red, which means they're dumping raw sewage into the rivers. And that's what happened on Monday. And we can expect that it will happen again in a matter of days in Philadelphia. Okay, and so I just want to thank a fantastic TA, Izzy Finkler, and the ecologists in my department to help me learn ecology, and others from the city who have helped with some of the other information.
Thank you, Jen. That was wonderful. In our remaining time, the plan that Anna and I came up with is that we would have question and answer, and hope for essentially discussion amongst all the participants. We've got lots of participants. Obviously, our panelists are still here. Anna, I see a couple of questions in the Q&A.
Maybe we can ask our panelists to turn their cameras back on so everybody can see them, if that works. I like that better than the boxes. So yeah, do you want to start with the first question, Ed, that was in the questionnaire, I think for Thomas?
I actually only see one, and it looks like it's been answered. I'm not sure how to work.
He wrote it in, he answered it, but we can talk about it. So someone asked you, Thomas, how you found your students were able to learn R, and if you had to make any modifications with that. So maybe you could comment on that.
So yeah, so they learned it better than I did, but I didn't have to practice as much. But what I found with the R Studio is that's tricky, because when you talk about inclusiveness, not every student was able to do it because they were using Chromebooks or they couldn't do it with the software downloaded to the computer. So in the groups of 4 or 5 or so students, at least one in every group was able to do it. And then I had Natalie come back and give tutorials. So during one Blackboard session, I had one of my students share their screen to troubleshoot some of the issues with running the script and so on. And she was able to show them, and then I recorded it so they can go back and watch it asynchronously. So for the future, we're currently working with our summer high school students, and we're looking into free online software like Galaxy and some other softwares that could do similar analyses, but hopefully a little bit more inclusive, that everyone could do it because they don't need to download that intensive R Studio software.
I guess, okay if I ask a question, Ed, real quick? I have a question for Alana and Isha, because I love sending the packages home to the students, but then obviously everybody's going to ask the question there, you need funders to fund that, right? And so do you have any ideas of how some way we as a community can do this? And I'm not saying without having funders, but maybe recycling the stuff, because I guess I'm trying to think, if can eClose people then go on to become developing future biologist people, and they might already have some stuff? Like, is there an encouragement for them to keep stuff, to share stuff? Are they asked what they might have? Or how can we then spread this to people and to other programs that may not have the resource to send out packets to everybody?
So every participant in eClose programs keeps their whole kit. And depending, they're iterative programs. So you start with level 1, level 2, like that. Yeah, we had level 3, and then we're like, oh no, we need a level 4. So many students want to come back. And for each kit, it's a set, dollar amount that they get of equipment in each kit. But then when they participate in the next program, that adds to that kit. And so in some of our programs, like the one we run at Fox Chase, we ask them to bring it all back because it wasn't for them to keep. And so we'll reuse all of that equipment for the next round. And then the students who have all the equipment at home, yeah, they can use it for everything. They have pipetters, they have little stereo microscopes, little tiny things. Yeah. So far we've provided funding, not us directly, but universities are sponsoring students who are incoming into college to take these courses, particularly if they're from schools that don't necessarily prepare them well enough for success. And that's been really, really a great way to do it. So universities who have a little bit of grant funding will sponsor 2 or 3 students. And I think that's really sort of the best model. So you see it works, right? And then we go to NIH and say this works. And then we just work together as a community to try to make every single student entering college have had a research experience before getting there.
Yeah, our experience a bit similar. Like, we also designed the package, and for the microscope and stuff like that, which is reusable, we provide them resources that even after your lab ends, you should try to do with this and try to share with your family, just to keep their enthusiasm throughout, even after the course finishes. And then we try to minimize the plasticware that we are sending them. So we have 6 to 7 labs. We try to put the minimum amount of stuff that can be shared between different labs. But yeah, that's a really good point. We also hope to collaborate with other people who are doing that, just to figure out if it can be shared with the community.
Yeah, I mean, it would be great if you could come up with a common dev bio kit or something that could then be used for multiple different, institutes or different sort of summer projects or different things. That'd be great. It's kind of also like those packages you buy now for your kids of meal preps or all those little things come in the mail every month. But
another way to do it is for a university to buy the first batch, to buy enough for a lab, right? And then we can still provide the programming and the annual reagents, right? But the cost gets prohibitively small, teeny tiny compared to what it is now. Yeah.
So I think there was a question. Do you want to take the one for Isha, Ed, in the Q&A?
Sure. Isha, there was a question concerning your tracking of students after graduation, and if you have begun that or been doing that, and just curiosity about, how they progress moving forward.
Yeah, so we keep in touch with our alumni even after the course. And we know like many of our alumni, like we have a number for the 2015 to 2019, like the people who participated in 2020, many of them still have to finish their undergraduate degree. So we know like 10 DFB alumni from 2015 to 19, they participated in the summer research program at other schools. And 2 of them actually applied to the University of Michigan prep program. And we know like that 6 of them are the PhD student at different universities, including UC Berkeley, UCSF, Wisconsin, and UPenn. And we have been trying to develop better methods to treat with our, to keep connected with our alumni. Now we have focusing on these long-term mentoring programs so that we can follow them throughout their career to see they have all the resources, not just one course to achieve what they want to do.
Isha, I had a question for you based on your talk. I was curious, and really perhaps for other panelists as well. How are the students that enter your programs? What are your criteria for choosing these students? What sort of metrics are you looking for?
Yeah, so we consciously reach out. So we have this huge list of different colleges, community colleges throughout the Puerto Rico and the Michigan. So mostly when our organization started, the 2 of the founder members, they were from Puerto Rico and they wanted to do something for the students there. So most of our students in the course, 50% are from Puerto Rico. So over a period of 5 years, we have compiled this huge list of the schools. So for every course year, we try to communicate to as many schools as possible, and community college, tribals, HBCUs and all the faculty working there, and encourage them for their students to apply. And when we receive the application, we just try to figure out what stage the students are there in career. We have a rubric of 5 points, whether this course is going to help them, whether they have enthusiasm based on their application for the developmental biology, and then do they already have enough research experience that they do not need this course. So we want to provide this course to people who are underserved basically, and who do not get the opportunities, maybe because of financial reason or any other reason. So if the people are working at a big university, we will generally not take them. We will prefer people who do not have any research experience, or who are working at the short, small place where there is no opportunities for such experience. So we keep all these things in mind.
Okay, we have a question that just came in that some panelists are typing to, but I'll ask it out live. So I'm inspired by your programs. Anyone can answer. I see the focus is on undergrad and community college students. Do you think these programs can expand to high school students or even younger? I guess I can start with Alana, because I know you started with high school students first for your program, kind of.
Yeah, our program started with juniors and seniors in high school. Largely because Fox Chase had a age limit that you couldn't be in a lab until you were 16. But now that we branched out into a nonprofit, we're kind of unlimited in terms of age. So we have students ranging from entering 6th grade all the way through retired adults who are interested in changing to biomedical jobs. So yeah, it's an approach that people can kind of dial into, and the younger students have amazing ideas. They can actually tell boy and girl flies just by looking at the vial. It's amazing.
Jen, do you want to answer verbally? I know you were typing stuff in, but
yeah, I was just going to say that I did actually share the DBH lab idea with a local middle school science teacher, and they used it and found the biomass of their trees. There's actually a ton of great resources out there that you can use, particularly if you live in or near a large urban center, because there's a lot of initiatives to map trees in cities. And so there's prefabricated ones, or you could just kind of come up with your own map like we did.
Yeah, Alana's comment was interesting to me, because actually I took flies into my kids' kindergarten classes, and the kindergartners can sort male and female flies. And it was kind of amazing to see. So you can go really young with these, and they get amazing questions, right? And they can ask things. Okay. Isha, Thomas, do you have anything to add to that one? Do you think you can expand your programs, or can you see it?
Yeah, for us, so far, we have just focused on the undergrad, but in the futures, maybe it's just because we invite them at the university of, so before the 2020, we used to invite them in the university to work in their lab. So we also had the age restrictions on the people, like what age people can enter the lab to work. But yeah, definitely in the future, in the virtual format, we can think about that.
Yes, and my work that I'm doing with the capstone students in my cell bio class, well, I'm actually doing it this summer, where I took on the West 11, I took on two a college now CUNY Research Academy students who are sophomores going into junior year, and they're doing research bioinformatically analyzing genes, differences between ovaries and testes of echinoderms, and they're using that whole bioinformatics computational biology approach. And I plan on exploring further expansion of this for using this for training high school students as well, and trying to get them to go from high school into LaGuardia to get their associates in biology and have more of a bioinformatics, biotechnology experience as well.
Thomas, it sounds like your work is sort of ripe for the development of case studies, which could be used, anywhere at many different levels, potentially cure courses as well. And I wonder if you've thought about that in any way. Really, the same question for Jen. I sort of fear going out and measuring trees because I've never done it. But if there's case studies that you know of, or things like that you're interested in writing, I'm just curious about your thoughts on that.
No, I never thought of that. This is just new that I started in the fall because of the pandemic. Otherwise, I was trying to always do the wet lab at LaGuardia, but this is something to look into for the future, doing a case study with this.
I think that's such a great question, and I'm smiling because I am a developmental biologist, right? So this is one of the furthest from the types of techniques that I'm familiar with. I work a lot with planarians. I was trained with zebrafish, C. elegans, and so it's very unusual. But as somebody who teaches intro bio labs, I need to have a lot of latitude. Not only that, I think it's silly to think that these fields don't really intersect. Ecological developmental biology is a really, really fascinating area. I have a whole section where we look at the intersection of environmental effects on mobility in C. elegans. In my lab, these are all types of things that are really interesting, and they're the types of things that when I talk with them about other people, they want to hear about them, and they'd love to try something like that. And I think that one element when it comes to kind of like doing case studies or like publishing about any of these things, one element that I think a lot of scientists would use that would be really, really helpful to get this type of thing into the publication arena just for sharing, would be to have some kind of common evaluation, some kind of assessment that would be applicable for any of these types of things. Now, I've been kind of diving into the social psychology research about education over the past several years. That's kind of like where my direction of interest has taken me. And so I've learned a lot about it, but I think it would be really great if we had some kind of ready to go open source tool that we could just use to see, like, did this element, how inclusive was this particular element? Like how effective was it at allowing students to see what they could do with this tool in the future, like in a job or something like that. These things that we know are associated with persistence in STEM. And so I think it would be really great if we had something like that. So it would make it so much easier for those of us who are doing things to just go out and publish it and share.
Okay, so I have a question for all the panelists now. One of the things we had selected everybody to speak at during this workshop was because you all did transition from one extreme to another during the pandemic. And one of the things we're interested as part of the Professional Development and Education Committee of SDB is sort of personal lessons that people have learned through this fast implementation process or switch during the pandemic, good and bad, right? So, I mean, we know that there were hard times, but there were some good things that came out of this. And Nicole had held a theme table yesterday. What's the silver lining to the pandemic? So I'm wondering if you could each kind of briefly reflect on what were some of the good and bad aspects of the implementation during this online virtual year, and what you see yourselves really continuing on with, right? That's something that came about through the pandemic. So what's the silver lining here? Why don't we start with Alana? We'll go in the order of the talks for fairness.
Yeah, I mean, COVID hit everybody like a hammer. We were actually running our in-person program. We were in week 8 out of 11 when we had to close the lab because we had 2 positive cases in Bucks County here in Pennsylvania, which were early cases in the pandemic. And, you know, it was kind of like a shock to everybody. The students were like, no, we'll still come. It's okay. We'll still come. And as scientists, you have to say, this is not safe, right? Like, this is not going to happen. So those students had to wait a whole year until we figured out how to do, Dara figured out how to do the online transition. And so we took a couple of weeks of, you know, watching everything go down, and then said, you know, all of these kids everywhere are going to lose the opportunity to become scientists. And so what can we do? So Zoom came out, you know, Zoom came big. And then, yeah, Dara figured it out. Like, she figured out exactly how to buy these tiny little, you know, professional lab equipment and how to fit it all in a 10 by 10 box. She is, like I said in the acknowledgements, a curriculum genius. Like, she can turn anything into something that's engaging and powerful, and with that kind of ownership. So I think we've really had overwhelmingly positive experience, and the concept of having kids from Kentucky working with kids from Texas, working with kids from Michigan and Washington State, you know, in this politically charged climate, like, these kids know somebody from those places that are their enemy on their local TV station. So the social impact has been mind-blowing, really. In terms of negatives, there are a lot of students who get dead fly or take the bag that says room temperature and stick it in the freezer. And so then, you know, there's a little bit of a pivot as soon as we get there, like, oh, all your flies are dead. But for many of our programs, we do it cooking show style. So we've set everything up, frozen the samples, and send them that so they can collect the data anyway. And so then it's kind of a learning experience of, you know, animal husbandry, things like that. But yeah, I mean, maybe Dara would have some more small negatives, but overwhelmingly, it's been something that really lets us know how much power it is for a student to really own their own research station and say, this is mine, I can research whatever I want.
Isha?
Yeah, for us, like, one of the important thing was that for the virtual course, we can enroll more students, because when we invite the students to our university, it is more costly because we also have, like, it's a free course. So we have to arrange for their housing as well as food. So we could take only like 20 to 24 students, but because it went virtual, we took like 36, 37 students for the course. So we were able to like reach out more and help more students. That was definitely an advantage for us. Also, we provide conference award to the students. So earlier the conference award were also expensive, but now like the conference award, because the conferences become virtual, the award also went less. So we provided like all our alumni conference awards and many of the students to attend the STB this workshop also. The other thing was really important for us, like earlier we were holding these panels which were in person. So we used to invite like local people as a panelist. So I think there was like, although we try to invite the diverse people, but we were still limited in that, like depending on who all are there in the nearby location. But because of the virtual format, we can apply, like we can ask people from like different places to be our panelists, which really like increased our diversity. So that was a really nice thing. And one more thing was that we were able to send the course supplies to students. So earlier they were visiting the university, but then they left, then they don't have any microscope or anything like that. When we are sending the course supply, they have everything with them. So like, you know, they can continue their exploration. Like if we are able to like sufficiently excite them about the developmental biology, they can continue on their own, like observe all these things in under the microscope, which is or fold scope, which is like really exciting. So that was like the good thing. I think the bad thing was like we also faced, like as Alana mentioned, some of the difficulties in the lab. We tried to send planaria and like some of them died because they were taking long in shipping or stuff like that. So some of us, our students like got worried, and we try to like calm them. That's okay. It doesn't affect your performance. But I know if I'm a student, I think I would be like really like got like sad by that if that happened to me. And I think there was a little bit like we tried to be interactive as much as possible. We had different like networking sessions and games during our virtual course, but I know that there's a lack of that personal interaction. So even some of the students are shy and they don't feel like talking to you. So if meeting them in person, you can you can try to engage them even if they're shy. But in the virtual format, like we said, like it's your choice whether you want to turn on the camera or not. So we feel like there were many students who did not talk during the course. So I think that was one of the bad things. And other thing was that for the earlier course, like for the 2015 and 16 the course, we went to Puerto Rico when the course was started. But after that, we started inviting the student to the University of Michigan, because we thought like when the students are coming to the university, they can actually see what a laboratory looks like, or what is a university like, how what different departments are there, which is one of the source of their excitement. But when they're working at home, it's still they're just room. So you cannot convey that excitement of being there in the laboratory or handling the pipettes for the first time, or seeing the thing with all the stuff that is present in the laboratory. So I think yeah, that's all the bad
You
things.
need like lab TikTok videos or something. So like around the labs.
Yeah.
Yeah. Thomas, what do you think?
So my negatives, I'm going to start off with first, so I can end on a positive note. I think I have a lot more positives than negatives. Number one is I actually never use Zoom for my teaching. I use Blackboard and the Blackboard Collaborate. And so the technical difficulties, so and then that's the biggest negative. Also, I don't require the camera on and so on. And a lot of times I think one of the aspects of being all online is, and this is for lecture and lab, is a lot of the times the students try to multitask. So some of them I've gotten that they're actually listening to me when they're in the doctor's office, or they're driving in their car, hopefully not as the driver, but as a passenger, and so on. So that distraction was a negative, really. And just like the students not having the appropriate software or hardware and the right computers and the lag time with internet connection. So I guess a lot of my negatives were the technical difficulties. On the positive side, I used Alice in Wonderland in my speech, now in my talk. Now I'm going to go and use The Wizard of Oz, right? Where you don't really have to look any further than your own backyard. And so one of the positives was I got to see how lucky and privileged I am to be working with the Wessel Lab and having that strong collaboration that I could use during the pandemic, but also continue to grow in the future, where this online component becomes a real godsend in a sense, in that now I could, once I get back to the wet lab, I can have them thinking about science after they leave the four labs of the walls of the lab, and they can bring this bioinformatic connected to wet lab. And with the online stuff of Zoom and so on, I can have Brown, my collaborators at Brown, talking with my students. They could do guest lectures. They can show them what they do at Brown with micro injections and so on. And then we could bring in the bioinformatics. So I still have that to leverage. And I can now outreach, I think, more to high school students and even citizen science and the community. And actually, not just what I was focused on for a large part of my career was the pipeline of strengthening transitions from associate to bachelor's to advanced degrees. But now I can look at the reverse and look at how can I strengthen the other end of the pipeline of taking the high school and teachers and helping strengthen that aspect, so I can get more students going for an associate to move them to a bachelor's in that, and really then extend at both ends of the educational spectrum the the increasing the participation of students in STEM, leading to advanced careers in STEM. So there's a lot of positives. I can go on, but I'm going to shut up. But yeah,
I think a question just came in, or a comment from Nicole Theodosio, who's our chair of the PDAC committee at SDB. And her comment is, it would be great to form more collaborations like Thomas has between R1 labs and PUIs of community colleges. Maybe something we can do together as a society. And I fully agree. That was actually going to be my question to Thomas, because there are so many things that the R1 institutions have that just PUIs and community colleges don't have. And forming just these individual lab and instructor or lab connections between people can really expose a large number of students to things that they're going to miss otherwise.
And I have to caution in that is the connection and the collaboration needs to be bi-directional. It can't be one way, because I'm a really strong advocate for the autonomy of community colleges. And a lot of times, whether we don't have enough faith in what we're doing and esteem and so on, or the other end, they might say, oh, well, they're community college faculty, they don't have a lot of research experience. It has to be both ways. You have to see the strengths in each other.
I 100% agree with you, because as I'm at an HBCU and I constantly get people wanting things from me, which is usually they want me to send them students, right? And I'm like, okay, there has to be an equal thing here. We're doing great research and stuff here too. So what can you do to help us? It's not just me kind of helping you sort of thing. And so I agree wholeheartedly, but I think it's something that the SDV should look at, and maybe we can help facilitate or make connections between labs at different sort of institutes, so we can expose a larger number of students to this topic that obviously we're all fascinated about. Okay, Jen, same question, still goods and bads.
Yeah, so I guess I'll start with the bad too. I think that it was kind of highlighted before. It's so easy to kind of lose students, and for students to lose themselves on through Zoom, right? So student engagement can be really not great. And one of the biggest concerns I would have about that, particularly in a large class, I have a total of about 100 students, is in the Zoom space, it's very easy to get completely lost and get so far off track that it's difficult to get back on track. And this is particularly concerning for credit-bearing semesters for college students or high school students or what have you, because there's really no system to be able to catch that. All of the things that we normally rely on are not there. And the other thing about it too is that it's often our most vulnerable students in that position to begin with. And so that I think was one of the more troubling aspects of the past year, which for me, the last 2 semesters were in person. 75% of the students were in person. But of course, there were space limitations in the laboratory. Normally I would have about 26 in the lab at a time, but I was only able to have 13 because of space limitations because of COVID. And so that meant they were only coming every other week to in-person lab. And then they were doing virtual lab on the other weeks. And it just became very, very technically difficult to do. I think I can also speak to a couple of things that were very helpful that I already had implemented in my class that made it really easy to go right into Zoom or go into an every other week in-person schedule, which is one thing is that I had flipped my lab class. So instead of doing a lab lecture followed by lab, I had already flipped it a few years before that and done recorded lectures and saved the in-person time for active learning, engagement, group work, that kind of thing, and lab stuff, right? And so that was really, really helpful because I could just give that to them. And then the other thing about it too that was really helpful is a lot of the labs that I've developed do require a lot of computational work. And so you'll extract DNA and send it for sequencing, then you have to analyze the sequence, or you have to design primers, or there's a lot of elements of that are real elements that real researchers are doing all the time, sitting there at the computer in the lab anyway, that were already incorporated into my class. I just had to figure out a way to cluster them all into one class, the virtual day, and then the in-person day would be different. And so, so I think that was really helpful. And a lot of other panelists also spoke about using bioinformatics and things like that, which we've done a lot. And those tools are so valuable, right? They're so important for researchers, and they're very much part of the research experience. And then maybe, I don't want to end on a negative here, but I think that there's something really special about the lab space that you don't necessarily have in a lecture space, it empowers students who might feel intimidated in lecture. Sometimes it can be more intimidating for some students too, right, to be in the lab space, but often the nature of the lab space is created for mistakes and for failure, because that's normal, and hopefully that's something that we convey in all of our lab classes, because if we if we expect that everything's going to be perfect, no one's going to drop anything and everything, like, right, no lab instructor expects that. So I think that there's something really special about the lab space in terms of creating an inclusive environment and a welcoming environment where students feel like they can belong. STEM identity and science identity is also another area that I've been really, really interested in. And I think that there's something about the lab space that can help incorporate more students into engagement and those wow moments. And that's something that we just did not have. We just did not have that on Zoom. And so there's a little bit element of mystery there. I'm trying to figure out what's how to make that happen in Zoom, but maybe we don't. Maybe things will be okay.
Yeah, I think your comment on failure is an important one, because I think students need to realize that it's not all right cookie cutter experiments, right? Real science doesn't work that way, and failure is okay sometimes, right? Sometimes experiments don't work, and I hope that the online labs we do can at least replicate that part, right? Because there's some element of learning in failing with something, right? Like it pushes you and the students above.
I had a question. I do want to make a comment in the chat. Ida, who, if I scroll back, she indicated that she thought NSF had some programs to facilitate interaction between R1 and other, so PYs and community colleges and such. And then there's some further information about that. And I know that Nicole's talked about this for a while now. So there's some folks at STB that are interested. So I hope that if there are people in the crowd listening, that they might reach out, maybe to Thomas, maybe to Nicole, whomever, to say, let's start a conversation about this. My last question, we're getting closer to the end. My last question kind of circles back to something that Anna asked early on regarding the kits that get sent out. And I'm thinking about Jen's use of sort of anything to measure a tree. I really liked that, because, why do I have to have some specialized piece of equipment? And I'm wondering if the folks that are actively sending things out, if they would envision being able to engage more students if they were only sending out the live specimens and the students were required to sort of come up with the other stuff. For Jen, it was at least the measuring tape sort of thing. But I was at a STB conference when we were still together, a handful of years ago, and there was a poster on building and using microscopes from mobile phones. So there's things like that out there, and I just wonder if if the kit, I'm making up a number, if the kit costs $75 with all inclusive and shipping included, could that number drop substantially if it was just the live creatures? Or shipping is kind of set and such, and we're not going to save that much money. I just wonder if that's something that might be worth exploring.
Yeah, I think exploring anything that could engage more people would be really fantastic. It gets a little bit complicated because, at least in Philadelphia, there aren't a lot of teachers who have any microscopes. So most of the high schools, middle schools, elementary schools in Philadelphia have no microscopes, like not one in the school. And so our experience with these students is that they go to college and they have no idea what anything is. So their ability to adjust to that environment where everybody knows what's going on except them is really not high. So we decided to do this with real lab equipment, just tiny, for that specific reason. But there are probably plenty of places where the students have microscopes in school. And one of the things we're going to be expanding out this year is our classroom program, where we've been basically collecting donors who give $1,500 for a classroom to get basically build the classroom into a research lab. And then they will only need the flies each year. So that's at a cost of less than $3 a student by the time we pay our little, students to prep the stuff. And so that becomes really feasible even on a $200 a year budget, which is what the Philly teachers have. So we want to do both things, like make sure that the students have professional equipment so that when they enter college, they are ready to go. Or if they're already in college and they can't get a research position, which is many of our students who we get, then they get the kind of experience where they can go in and tell the PI that they're interviewing with, I got this, I've done this before. So I think we have to maintain both things, and the concept of having, more well-off people with grants, pay for each of the processes is really, that's where we're going.
Yeah, to add to that, like for most of our students, we also try to take them from the underserved background. So they already have like limited facilities. So we try our best to provide them everything. So they have all the resources to do the experiment, because we don't want to make them think, I know like science is hard, but we don't want to make them like a false feeling that it is undoable. Because if you do this for the students, you don't have to provide them resources. It can create a false sense that I really can't do that. I'm not suitable for this, because they are at such a young age. So they do not have experience to understand that this is how science is done. The other thing, for our students, we want to, so most of our students have like no previous experience in the lab or research. So the problem is that because they have no experience, they are not, when they apply to the graduate school, they are not accepted. So we really want to make them competitive enough. So we really want to provide them like real hands-on labs. So when they apply to graduate school or any other program, they stand a real chance. But yeah, I know like one of the things like we supplied was Fold Scope, which was developed by Manu Prakash Lab, and it's like just $1.75 each piece, like which can be really good. Like those kind of things like can be like made available widely, which doesn't cost that much. Yeah.
And we had a question that just came in from one of the attendees. How do you start the conversation, whether you're reaching out to community colleges or high schools, to create a program like Alana or Isha have going? Is it as simple as writing an email? What are your comments?
No, I wish I could say yes, but no. So the problem that we've had, particularly with high schools, so the community colleges, they want to participate if they're going to get credit. And so we haven't achieved that yet. So Fox Chase is a standalone cancer center. There's no education credits. There's none of that. So we haven't been able to, create a course that people get credits for. And so why would they do this when they can get the credit? So that's one problem. And then the second problem is that the teachers are so overwhelmed by this No Child Left Behind curriculum that's oppressive. And so how do they fit in something that's completely brand new, especially in a place like Philly, where the magnet schools in Philadelphia, everybody on earth who wants to try out their new science thing goes there. Like, we want your underserved kids to prove that our thing works, and they only go to the magnet schools. So now you have like 98% of the students of Philadelphia who get nothing, right? And then the magnet schools that get everything and get to pick and choose which programs they want to try. So just reaching out to a teacher and saying, I have this great idea, you could try it, but if you don't have a really close personal relationship with them, it's not going to be easy. So the way Dara decided to do this at the beginning was to bring teachers in and train them to be scientists first. So we do that at no cost to the teachers, and then 40 to 60% of the teachers the following year will bring the program into at least one classroom. So that's how we've done it, is to actually train the teachers and then send them back. And yeah, like I said, it's sort of like a 40% a year kind of hit rate, but then a lot of the times the 60% who didn't do it will come back, right? I'm thinking about this, how do I integrate it? So one of the most important things if you're going to do high school is to make it curriculum aligned, so that the teacher can check off, like, I met all of these criteria that's mandated by the school district. So that is absolutely critical. If you want to talk about it, just let us know.
Yeah, and I know I didn't describe a program like that, but I've worked over the last 15 years or so with a lot of schools in Philadelphia, one through an extension of the BioWise program, actually, called Bridge to Rebio at Penn. And some of the schools that we worked with were incredibly under-resourced. It's great when you can have a college, like a program at a large RU or something that is interested in doing outreach to those schools and not to like the magnet schools, because it's really, it can be really transformative for the students. I actually know some of this, like there was a student at West Philly High who I had as one of my mentees, who is now, like, I know him now still as an adult with, and it's really great to see that career trajectory. But I think that that relationship with the school is the most important thing. And it's usually a person at the school. It could be a person in the administration, but more likely it's going to be a teacher. And you really need to have that commitment, because without that, it's not going to be sustainable. There's so many other barriers that they're already dealing with that it's just not going to be it's going to be too much to even approach a school to do something like that. Also, the curriculum is usually available online. It's really easy to find typically for most states. And so, so usually you can go ahead and just check that off. It's really funny in the biological sciences, like in Pennsylvania, a lot of the curriculum is just like, you're like, where does this fit in? Like, I know like planarian regeneration, for example, like it's very difficult to parse out because the nature of the wording of the curriculum is not what we would expect as academics in the field. But it's also very insightful to look at.
Okay, everyone, we've reached our limit, so I want to take an opportunity or a moment to just thank all of our speakers, thank my co-chair Anna, and our committee chair Nicole, and of course Ida. I'm not sure if she's there, but I can see her. I can't see her picture, but she's at least around. So we're going to go ahead and end our session.
Round of applause for everyone so you can hear.
Hi, Ida. Thank you. So we're going to go ahead and end our session, and we hope everybody has a great rest of their day.
Thank you, everybody, and keep on doing your great jobs. So nice to see you all.
Mhm.
Bye.
Thanks, everybody.